Okay, let's unpack this.
Welcome back. So last time we really cemented Mendel's foundational laws, didn't we? Segregation, independent assortment.
Right. How alleles get passed down gave us those nice, clean F2 ratios, 3.1, 9.3.3.1, textbook stuff.
Exactly. The rules of transmission. Simple, elegant, almost.
Almost. Because, life gets complicated. Those perfect ratios, often not what we actually see out there.
Precisely. So today we're shifting gears a bit. We're moving from just how genes are transmitted to how they're actually expressed.
Ah, okay. So beyond simple dominant versus recessive.
Way beyond.
Yeah.
Our mission today is really to dig into the necessary extensions and modifications of classic Mendelian genetics. We need to look at situations where expression isn't straightforward.
Like how the gene product itself behaves, or maybe how multiple genes get involved.
Both. And also where the gene is physically located on a chromosome, and even, the environment playing a role. It all feeds into the final phenotype.
Gotcha. So the goal for you listening is to get why that simple 3.1 ratio is, frankly, pretty rare in the real world, and how all these complex interactions really shape what an organism looks like and how it functions.
Let's start with the basics, the alleles themselves. We know they're different versions of a gene, right? Arising from mutations.
And we usually talk about the wild type allele. That's the common one.
Generally, yes. The most frequent one found in a population, it's often dominant, but critically not always. Then you have the mutant alleles, the ones with changed genetic info.
And these mutations actually change what the gene does, the function of its product.
Exactly. A lot of the time, maybe most often, a mutation causes a loss of function. The protein or enzyme it codes for just doesn't work as well, or maybe not at all.
And if it's completely non-functional?
We call that a null allele, complete loss.
A null allele. Sounds pretty definitive. Is that usually behind the more severe genetic conditions?
Often, yeah, that makes sense. But sometimes you get the opposite, a gain-of-function mutation.
Okay, how does that work?
Well, the mutation might make the protein hyperactive, or maybe just makes the cell produce way more of it than usual.
Ah, and that often leads to a dominant phenotype, right? Because even one copy doing too much can mess things up.
You got it. Think about cancer. A normal gene-regulating cell growth, a proto-oncogene, gets a gain-of-function mutation and becomes an oncogene driving uncontrolled division. That's a classic example.
Okay, so that's the alleles themselves. Now, how do different alleles for the same gene interact within one individual? This is where we really start seeing those Mendelian ratios change.
Right. First up, incomplete or partial dominance. This is a big one.
That's the snapdragon example, isn't it? Red flowers crossed with white flowers.
Yield F1 offspring that are all pink. The phenotype is intermediate, a blend, visually speaking.
But genetically, it's not really a blend, is it? The alleles are still distinct.
Absolutely not. It showed early on that blending inheritance wasn't the whole story. The alleles, and say, are still discrete. But because the heterozygote looks different from either homozygote or $2, the F2 ratio changes dramatically.
Okay, so instead of 3.1 red to white, what do you get in the F2?
You get a phenotypic ratio that perfectly matches the genotypic ratio. 1 red, 2 pink, 1 white, 1 white.
Why pink, though? What's happening at the molecular level?
It's often about dosage. The pink heterozygote likely produces only about half the amount of red pigment compared to the red homozygote. Less pigment means a lighter color pink.
That makes sense. It reminds me of the threshold effect you hear about with some human diseases.
Exactly. Take Tay-Sachs disease. If you're heterozygous, you have one working allele and one null allele. You only produce maybe 50% of the needed enzyme.
But that's enough.
It seems to be. 50% is above the minimum threshold required for normal function. So the heterozygote is phenotypically normal, even though biochemically they're different.
Okay, so incomplete dominance is this intermediate phenotype. What about codominance then? How's that different?
Codominance isn't about blending. It's about both alleles showing up distinctly and equally.
Ah, like the MN blood group in humans.
Perfect example. The LN-LNL heterozygote doesn't have an intermediate blood type. They express both the M antigen and the N antigen on their red blood cells. Both products are fully formed and detectable. No blending, just joint expression.
Right. Okay, so that's interactions of alleles for a single gene. But things get even more complex when you consider that in a whole population, there can be more than just 2 alleles for a gene, right?
Definitely. We call that multiple alleles. An individual can only carry 2, of course, being diploid. But within the gene pool of the population, many different versions can exist.
And the classic example here has to be the ABO blood group.
It's the best one to illustrate this. You have 3 main alleles: IALO, $10 and $10.
And the dominance relationship is interesting too. Ida and Ella are both dominant over ILO, but they're codominant with each other.
Exactly, leading to the 4 blood types. Type A, IA, IAIA, IEA, type B, IBA, IAII, IBA, OEE, IBA, and type O.
But what's really cool is the biochemistry underneath, this H substance thing.
Oh, absolutely. It's a fantastic molecular story. The H substance is basically a precursor molecule, a carbohydrate, sitting on the surface of red blood cells.
And the ABO gene determines what gets added to it.
Precisely. The enzyme coded by IAA adds a specific sugar, N-ethyl-galactosamine. The IBAB enzyme adds a different sugar, galactose, and the IVAE codes for a non-functional enzyme, so nothing gets added.
So type A people add the A sugar, type B add the B sugar, type AB add both.
And type O add neither, leaving just the basic H substance exposed.
Which brings us to that really mind-bending case, the Bombay phenotype. How can someone be genetically, say, type AB, but test as type O?
Yes. This is a beautiful real-world example of epistasis, which we'll talk more about later. The Bombay phenotype comes from a mutation in a completely different gene called FUT1.
And what does FUT1 do?
It controls the synthesis of the H substance itself. If you have 2 recessive mutant copies of FUT1, genotype HH, you cannot make the H substance precursor at all.
Whoa. So even if you have the IAA and IBANO alleles.
Exactly. Even if your ABO genotype is EA-IBE-BO, the enzymes produced by those alleles have nothing to add their sugars to. The H substance isn't there.
So on a blood test, they lack A and B antigens and look like type O. Wow. The FUT1 gene completely masks the ABO gene expression.
It's a perfect illustration of gene interaction. And this idea of essential genes leads us to another modification, lethal alleles.
Genes that are absolutely required for survival.
Right. A mutation in such a gene can be lethal. We often talk about recessive lethal alleles. You can carry 1 copy fine if you're heterozygous.
But if you inherit 2 copies, homozygous recessive, it's fatal.
Correct. The classic textbook example is coat color in mice, specifically the yellow allele, AY.
Yes, yellow coat color is dominant over the normal agouti color. But if you cross 2 yellow mice,
which must be heterozygotes, AYA, you don't get the expected 1.2.1 ratio of AYA. Instead, you get a 2.1 ratio in the offspring.
So 23 yellow and 13 agouti. What happened to the homozygous yellows?
They die in utero. The AYA genotype is lethal early in development, so those mice are never born, skewing the ratio of live births.
That's wild. And isn't there a complex molecular reason for that? The A allele does more than just affect color.
There is. It turns out the mutation causing the yellow color, which is dominant, is actually deletion, and this deletion extends into a neighboring gene called MERK, which is essential for development.
So the AY allele is dominant for color, a gain of function in a way, but it also creates a loss of function in the essential MERK gene, making it a recessive lethal. 1 allele, 2 very different effects.
Exactly. Complexity upon complexity. Now, what about dominant lethal alleles? If just 1 copy is enough to kill you, how can those possibly persist in a population?
Yeah, that seems counterintuitive. They must have a trick.
The trick is usually delayed onset. The lethal phenotype doesn't manifest until later in life, often after the individual has already reproduced and passed the allele on.
Huntington disease is the tragic example here, isn't it?
It is. Symptoms typically don't appear until middle age, long after many people have had children, so the dominant lethal allele gets transmitted generation after generation.
Okay, let's shift slightly. We often think 1 gene, 1 trait, but that's often not true either. There's pleiotropy.
Right. Pleiotropy is when a single gene influences multiple, often seemingly unrelated, phenotypic characteristics.
Like Marfan syndrome.
Yeah.
Where 1 faulty protein affects the eyes, the heart, the skeleton.
Exactly. A defect in the fibrillin gene causes problems across multiple body systems. Porphyria variegata is another example. Issues with an enzyme in heme production causing abdominal pain, neurological symptoms, skin problems. 1 gene, many effects.
This kind of complexity must make research tricky. How do geneticists figure out if 2 different mutations causing similar problems are actually in the same gene or different ones?
That's where a crucial tool comes in. Complementation analysis. It's fundamental.
Right. You need to know if you found a new gene or just another broken version of one you already knew about.
Precisely. Let's say you have 2 different true breeding mutant fly strains, and both are maybe wingless. You cross them.
Okay. What are the possible outcomes?
Outcome 1. The F1 offspring have normal wings. They are wild type. This means complementation occurred.
Meaning the mutations were in different genes. Each parent strain provided the working copy of the gene that was mutated in the other strain.
Exactly. The mutations complement each other. They fall into different complementation groups, meaning different genes.
And outcome too.
Outcome 2. The F1 offspring are still wingless, no complementation. This tells you that both original mutations must be alleles of the same gene. They fail to complement because neither parent could provide a working copy of that specific gene. They're in the same complementation group.
That makes sense. It's a clever way to sort out the genetics. And that leads us nicely into situations where we know multiple genes are affecting one trait. Gene interaction.
Right. Often, a single phenotype is the result of a whole chain of steps, a developmental pathway. Think of it like an assembly line that's epigenesis. Different genes control different steps.
And if one gene in the pathway is broken, the whole process can grind to a halt, affecting the final outcome?
Exactly. And when one gene's alleles mask or modify the effective alleles of a different gene, we call that epistasis.
Okay, so one gene messes with another gene's expression. There's a dominant gene and a recessive one in this relationship.
Sort of. We call the gene doing the masking the epistatic gene, and the gene being masked the hypostatic gene.
Got it. And this is where those weird F2 ratios expressed in sixteenths come from, right? The power of sixteenths.
That's the clue. If you're looking at a single trait code color, fruit, shape, whatever, and you do a dihybrid cross, and the F2 ratio adds up to 16 parts, like 9.3.4, 9.7, you've got two gene pairs interacting, assuming they assort independently, of course.
Let's walk through a couple of those. How does recessive epistasis work to give a 9.3.4 ratio?
Okay, think about mouse code color again. Let's say gene B controls pigment production. B for pigment, BB for non-albino. Gene A controls the pattern. A for agouti, A for black.
Right.
In recessive epistasis, the homozygous recessive condition at one locus, like BB albino, masks the expression of the other locus, A. So if a mouse is B, it's albino regardless of whether it has the A or A allele. It can't make any pigment, so the pattern gene is irrelevant.
So the ABB mice look the same as the AA mice. They're both albino. That combines two categories from the 9.3.3.1 into one group of 4. Makes sense. 9, agouti AB. 3, black AB. 4, albino AB plus A.
Exactly. Now, contrast that with dominant epistasis, which gives a 12.3.1 ratio.
How does that work?
Here, a single dominant allele at one locus, say A, is enough to mask the expression at the second locus, B. Summer squash color is a good example. If a squash plant has the dominant W allele for white color, the fruit is white. It doesn't matter if it also has the allele for yellow Y or green Y. White masks everything.
So WY and WY plants are both white. That lumps the 9 and the 3 from 9.3.3.1 together, giving 12 white. Then you have the WWY yellow 3 parts and DIBY green 1 part, 12.3.1.
Okay. Then there's complementary gene interaction giving 9.7. This one feels different.
How so?
Here, you need at least one dominant allele at both interacting loci to get the final phenotype. Think of sweet pea flower color. You need enzyme C from gene C and enzyme P from gene P to make purple pigment.
So you need to be CMP to be purple.
Exactly. If you're missing either one, like CPP, CCP, or CCPP, the pathway is broken and the flowers stay white.
So only the CP genotype 916 gets you purple. All the others, CPP, CCP, CCPP, combine to make up the 716 white category, 9.7. Clever.
And sometimes the interaction just creates brand new phenotypes, like the 9.6.1 ratio in summer squash fruit shape.
Okay, what happens there?
Let's say you have genes A and B. If you have dominant alleles for both, AB, you get a disc shape, 9-16. If you have a dominant allele for only one gene, maybe B or AB, you get a sphere shape, combining for 616. And if you're homozygous recessive for both, AB, you get a long shape, 116, 9.6.1.
Wow. So the interactions generate diversity. Now, you mentioned a twist even with the classic 9.3.3.1 ratio.
Yes, sometimes you see that ratio, but it's describing variations of a single characteristic. Drosophila eye color is the classic case.
Fruit fly eyes. Wild type is brick red, right?
Right. That red color is actually a mix of two pigments, a bright red one, Drosophila, and a brown one, xanthomatin, produced by separate biochemical pathways.
Okay. And mutations can block those pathways.
Exactly. The brown mutation blocks the Drosophila pathway so the eyes look brown. The scarlet mutation blocks the xanthomatin pathway so the eyes look bright scarlet.
What if a fly has both mutations? Homozygous recessive for both bound and cystidollars.
Then both pathways are blocked. No red pigment, no brown pigment. The eyes end up white.
So if you cross two double heterozygotes, Bond brown plus Blastus baller, you'd expect the 9.3.3.1 ratio, but it represents 4 phenotypes for eye color. 916 wild type, red, 316 scarlet, 316 brown, and 116 white, the double mutant. It's still gene interaction, just fitting the familiar ratio pattern.
That really shows how deep these interactions go. Okay, wrapping up, let's broaden out again. It's not just about allele interactions or multiple genes. Where a gene is and the organism's sex and its environment, they all matter too.
Huge factors. Let's start with location specifically, X-linkage. Genes on the X chromosome have unique inheritance patterns.
Because males only have one X chromosome, right? They're homozygous.
Precisely. They only get one shot, one allele for any X-linked gene. Thomas Hunt Morgan figured this out with his fruit flies.
The white-eye mutation, that's the one. He did reciprocal crosses. White-eyed female, X, red-eyed male, gave different results in the kids and grandkids compared to red-eyed female X, white-eyed male.
And that difference could only be explained if the eye color gene was physically on the X chromosome.
Exactly. It was landmark proof linking a specific gene to a specific chromosome.
And this leads to patterns like crisscross inheritance for recessive X-linked traits.
Right, where a trait goes from the affected mother, homozygous recessive, to all of her sons, because they inherit her only X chromosome.
But hang on, not all traits tied to sex are actually on the X chromosome, are they?
Good point. No. We have sex-limited and sex-influenced inheritance. These involve genes on the autosomes, the non-sex chromosomes, but their expression is modulated by the individual's sex hormones.
Okay, what's the difference? Sex-limited.
Means the phenotype is expressed in only one sex, even if both sexes carry the alleles. Think milk production in cattle. Bulls have the genes influencing milk yield, but obviously they don't produce milk. The trait expression is limited to females.
Makes sense. And sex-influenced.
Here, the trait appears in both sexes, but the dominance relationship is different between males and females, usually because of hormones.
Ah, like pattern baldness in humans.
A classic example. The allele for baldness acts like a dominant trait in males. A heterozygous man often becomes bald, but it acts like a recessive trait in females. A heterozygous woman usually doesn't lose her hair. Same genotype, different phenotype depending on sex.
Fascinating. Okay, finally the big one. The environment. The genotype isn't destiny, is it?
Not at all. The phenotype is often a result of interaction between the genotype and the environment. We need to consider penetrance and expressivity.
Define those for us. Penetrance.
Penetrance is the percentage of individuals who have a specific genotype and actually show any sign of the corresponding phenotype. Like if a gene for a dominant disorder has 80% penetrance, 20% of people with the allele won't show the disorder at all.
Okay. And expressivity.
Expressivity is the range or degree to which the phenotype is expressed in individuals who do show it. It's the variation in severity.
Got an example.
The eyeless mutation in Drosophila. Flies with the exact same eyeless genotype can have eyes ranging from perfectly normal to slightly smaller to completely absent.
Right.
That variation is expressivity. It can be influenced by genetic background, temperature, other factors.
And temperature itself can be a direct environmental switch, right? Conditional mutations.
Absolutely. Think Siamese cats or Himalayan rabbits. They have dark fur on their paws, ears, tail to cooler extremities.
Why?
Because the enzyme needed to produce dark pigment in their fur is temperature sensitive.
Ah.
It only works properly at the lower temperatures found in those body parts. In the warmer core body areas, the enzyme is unstable and the fur stays light.
Wow. The environment literally sculpts the phenotype. Nutrition can do this too, like with PKU.
Right. Phenylketonuria. A strict diet low in phenylalanine can prevent the severe effects of the genetic defect. A clear environmental intervention modifying a genetic condition.
We also see variation in when a genetic trait appears, the onset of expression.
Yes. Many aren't present at birth. Tay-Sachs. Huntington's disease manifest later in life. And related to that is genetic anticipation.
What's that?
It's a phenomenon where a genetic disorder appears progressively earlier and often with increased severity in successive generations. Myotonic dystrophy is a key example.
And that's often linked to what? Unstable bits of DNA?
Often, yes.
Yeah.
Unstable repeats in the DNA sequence that tend to expand from one generation to the next, worsening the gene's function.
Okay. So pulling all this together, modified dominance, multiple alleles, epistasis, location, sex, environment, timing, it paints a much richer, more complex picture than simple Mendelian rules.
Absolutely.
Yeah.
Mendel gave us the fundamental rules of inheritance, how genes are passed on. But all these extensions and modifications, they reveal the rules of expression, how that inherited potential actually translates into a physical organism, influenced by everything from other genes to the temperature outside.
Which really brings us back to that fundamental question, doesn't it? The ultimate interplay.
You mean nature versus nurture?
Yeah. I mean, listening to all this, it seems like every trait we've discussed, even the ones caused by a single gene defect, has some environmental component influencing the final outcome. The phenotype is always genetics plus environment interacting.
That certainly seems to be the case, from obvious environmental factors like temperature or diet to subtle ones like genetic background effects. It's always a mix.
So here's the final thought for everyone listening. If every single trait, from height and eye color to complex diseases, demonstrably involves both genetic predispositions and environmental influences, should we maybe consider that whole nature versus nurture debate finally put to bed? Or does the fact that the ratio of genetic versus environmental contribution varies so wildly for every trait mean the conversation and the research into that balance is actually more important than ever? Something to ponder.
Indeed, a lot to think about. Thank you for joining us for this deep dive into the fascinating complexities beyond basic Mendelian genetics.
A warm thank you from the Deep Dive team. We'll catch you next time.